Today I'm going to be reviewing topics covered in the Cell Specialization and Homeostasis unit. These topics include the cell cycle, mitosis, stem cells, differentiation, tissues, organ systems, plant specialized cells and tissues, homeostasis, and feedback mechanisms. The purpose of the cell cycle and mitosis is growth, repair, and replacement. In order for an organism to grow, their cells don't become larger. They duplicate their cells through the process of mitosis to create more cells. Repair, if you get a cut on your skin, your surrounding skin cells go through the process of mitosis to repair that cut. And of course, replacement, you're losing skin cells all the time. So through the process of mitosis, your skin is able to replace those that are lost. It's important to remember that one parent cell, which is the original cell, will make two identical daughter cells. So the cell cycle can be divided into two general parts, that is interphase and cell division. So look down at this picture. Interphase goes around the outside. It's the part that takes up most of the cell cycle. It's where the cell will spend most of its life. Then we have cell division. Now interphase includes three parts, G1, S, and G2. Here you can see interphase includes G1, which is growth, S, which is DNA replication, that's very important to know, and G2, which really is the final preparations for mitosis. Then if we look specifically at cell division, there are two parts. There's mitosis, which is the division of the nucleus, and cytokinesis, which is the division of the cytoplasm. So here we have mitosis, which is sometimes called the M phase. So it goes in this order. So the cycle is moving this way. So it goes in this order: prophase, metaphase, anaphase, telophase. Okay, and then finally cytokinesis. And then you have two identical daughter cells, which will then go back into interphase and start the cycle over again. So mitosis, again, includes four parts. We'll talk about those four parts briefly. So again, we have interphase, where the cell spends most of its life. You can't really see the chromosomes. They're not condensed. They kind of look thready. In prophase, you can start to see the chromosomes. They have condensed, and the nuclear envelope around the nucleus is disappearing. You also see the centrioles are moving to opposite ends of the cell. Metaphase is characterized by the chromosomes lining up along the middle and the spindle fibers attaching to the centromere of each chromosome, because in anaphase, those centrioles are going to shorten, the spindle fibers are going to shorten and pull the chromosomes apart and pull them to opposite ends during anaphase. And then in telophase, the nuclear envelope reappears for both of the two new daughter cells. And in animal cells, you'll see a cleavage furrow. And then finally, in cytokinesis, the cytoplasm separates, and those two daughter cells will go back into interphase and start the cycle again. The checkpoints in the cell cycle are really just to make sure that everything in the environment is stable and good for replication. It doesn't make sense for the cell to make more of itself if the environment can't support more of it. Is there enough space? Is there enough food, nutrients, etc.? And then it'll also check that things inside the cell are going well also. So during the M phase checkpoint, it'll check to make sure that all the spindle fibers are attached correctly to the chromosomes. In G1, it checks to make sure that cell size is good, that there isn't any DNA damage. In G2, it'll check again cell size, DNA replication, preparation for mitosis, etc. This is something very important to understand going forward. You can approach this from two ways. So starting with the complex organism, understand that complex organisms are made up of organ systems. Organ systems are made up of organs. Those organs are made up of different types of tissues. Those tissues are made up of specialized cells. They have a specific function. Inside the cells are organelles. Those organelles are made up of different groups of macromolecules, made up of molecules, made up of atoms. Or we can approach it from the other way, that atoms make up molecules, which make up even larger molecules. Macro means large. Those make up the organelles. Organelles are found inside cells, so you could say they make up cells. Cells make up tissue. A whole bunch of cells together doing the same thing makes up a tissue. The tissue makes up the organ. An organ is part of an organ system, and an organ system makes up the complex organism. So here we have an unfertilized egg. Fertilization takes place with sperm, and once the DNA of the sperm gets in with the DNA of the egg, we have the zygote. The zygote is the fertilized egg. It's a single cell. All multicellular organisms start out as a zygote, as a single cell. So after fertilization, the cell immediately begins mitosis. So remember that growth takes place through the process of mitosis. So we go from 1 cell to 2, 2 to 4, 4 to 8, 8 to 16, etc., until we get the morula, which is just a big ball of all the same cells, a little bit more division, and we start to see different types of cells. Because at this point, the cells have started to differentiate, which I'll talk about in a moment. But inside the blastocyst, we have the embryonic stem cells. These are very important. So a few important definitions to know. First, specialized cells are cells that have a specific structure in order to perform a specific function. So here are a whole bunch of examples of specialized cells. Muscle cells are specialized to perform a specific function. Blood cells are specialized to perform a specific function. Nerve cells, cardiac cells, liver cells, intestinal cells. I could go on. Skin cells, hair follicle cells. The list goes on and on and on. We have over 200 different types of specialized cells in the human body. How do those cells get to be specialized? How do they go from being a stem cell, which is an undifferentiated cell that creates different specialized cells? So it's undifferentiated. So how do we get from an undifferentiated cell to a specialized cell? And that is through the process of cell differentiation. And this happens through gene regulation. So perhaps there are certain things in the environment that will cause some genes to turn on and some genes to turn off, or perhaps one cell has more of a certain type of protein than another cell. That will cause genes to regulate. So in a muscle cell, even though the muscle cell has the same exact copy of DNA as, let's say, the nerve cell, they're different because different genes are being used inside those different cells. This just goes over different levels of differentiation. So in the zygote, we have the single cell, then that forms eventually the blastocyst, and inside the blastocyst are stem cells. The stem cells are pluripotent. Pluri means they can become many different types of cells. The zygote is actually totipotent. Toti meaning it can become any type of cell that is either in the organism or helps the organism develop. The stem cells inside the blastocyst can only become part of the organism. The more differentiated the cells become, the more destined they are to become a certain type of cell. They become less potent. They're less able to become other cells. So once a liver cell is destined to become a type of liver cell, we call them multipotent. They're more differentiated. So here are some examples of specialized animal cells and how their structure helps in their function. I suggest you pause here for a few minutes, look over these, study them. You definitely need to know them. So the most common example is the specialized cell red blood cells. Their function is to carry oxygen, and we want to maximize the amount of, not we, but our bodies want to maximize the amount of oxygen that those red blood cells can carry. So they have large surface areas to allow oxygen. They contain hemoglobin. It's a protein that the oxygen attaches to. It also contains no nucleus. It's like, why bother wasting the space with the nucleus when we need to get as much oxygen into these red blood cells as possible so that the rest of the body can have oxygen. So again, stop here, study these, think about how the features, the structure of the cell helps its function. Here we have some examples of plant specialized cells. So this is a root hair cell. Okay, it has these extensions which maximize the surface area. These extensions allow for more water to be absorbed into the root. And obviously that's good for the plant. The plant needs water. So again, take a few minutes, stop here, review how the structure is related to the function, because the structure is going to help in its function. That's why we have specialized cells. That's why they're different. So I talked a little bit about gene regulation. I don't expect you to know very much about this, other than the fact that in order for cells to become specialized, they're going to use different genes. You have over 20,000 genes, or about 20,000 genes. They're not sure of the exact number, but it's close to about 20,000. When different proteins are in the cell, different genes can be turned on and turned off. Or if the cell detects something in the environment, different genes can be turned on or turned off. And that, again, is how cells become specialized. Although they have the exact same amount of DNA, the exact copies of DNA in every cell, They're able to become different because they're using different segments of the DNA. It's called gene regulation. We learned about the 4 types of tissue in the human body. They are connective tissue, epithelial tissue, muscle tissue, and nervous tissue. Here, it goes over the function. So epithelial tissue, that covers the body and organ surfaces. It lines body cavities and forms glands. It's involved with protection, absorption, excretion, secretion, diffusion, and filtration. I'm not going to read through the rest of these. Take a minute to pause and review the different tissues. Remember, tissues come together to form organs. I want to review the fact that there are 3 different types of muscle tissue. There is smooth muscle, cardiac muscle, and skeletal muscle. And the reason I want to review this is to reinforce the idea that there are differentiated cells. There are specialized cells. So even though they're all muscle, they have even further differentiated, become even more specialized to do a specific job. Cardiac muscle in the heart is very different from smooth muscle or from skeletal muscle because it has a completely different function. The structure matches the function. Here we have the tissue of a plant. Specifically, I've put up a picture of a cross-section of a leaf. We can see vascular tissue. It's very important to understand that vascular tissue is made up of xylem and phloem. And here you see that xylem transports water throughout the leaves, and phloem carries food made in the leaves to other parts of the cell. And phloem can also carry water, but xylem cannot carry food. Xylem, the water moves in one direction, whereas the phloem, it can move in multiple directions. And again, if you look here specifically at the structure of the cells, you can see that they're long, thin, and hollow. The structure matches the function. The reason they are structured, the reason cells become specialized, is to help in the functioning of the organism. We want to be as proficient as possible. Then we went into organ systems. We learned about the musculoskeletal system, digestive, respiratory, nervous, circulatory, and a little bit about reproductive. I want to go over the relationship between the circulatory system and the respiratory system. So this represents the tissues of the body. So blood will flow to the tissues of the body from the heart. Okay? And it gives the oxygen to the tissues of the body and picks up carbon dioxide, which is a waste product of cellular respiration. We need oxygen for cellular respiration. We get CO2 out of cellular respiration, but that's a waste product which we will have to excrete. So the blood, now deoxygenated, flows back to the heart. Okay, so this side of the heart is deoxygenated blood, and its purpose is to pump the blood to the lungs. And why do we need to send our blood to our lungs? To become oxygenated and to get rid of the carbon dioxide that we no longer need. So carbon dioxide leaves the lungs, oxygen comes into the lungs, and oxygen diffuses across the surface of the lungs and oxygenates the blood. That oxygenated blood then comes back to the heart and is sent by the contraction of the heart out to the rest of the body, whether that's to the upper part of the body or to the lower part of the body. But that's essentially what happens with the blood and the lungs. The lungs are part of the respiratory system, whereas the heart, obviously, is the circulatory system. I apologize for my writing. So that is how the respiratory and circulatory system are related. Blood flows through and past the lungs to pick up oxygen. The circulatory system sends that oxygenated blood around the body for the main purpose of getting oxygen to the tissues of the body. The relationship between the digestive system and the circulatory system. So this shows the small intestine. In the small intestine, there are tiny structures called villi. They maximize the surface area of the small intestine. We need to absorb as much nutrients as we can. But through the villi run arteries and veins. So they're capillaries. And what happens is that as the blood flows through these capillaries, it picks up nutrients from within the small intestine. Okay, so some of those nutrients go straight through, some of them go into the cells and then are diffused across. But that is how nutrients get from our small intestine into our blood. And then the blood carries those nutrients around our body and provides those nutrients to our different tissues and our different cells. So how is something that I eat, how does the glucose in that get to the tip of my toe? Well, it does that by diffusing into the capillaries that are surrounding the small intestine, and then goes to the rest of the body through the blood. Another example of a relationship between organ systems is the relationship between the muscular system and the reproductive system. So the uterus in females has muscle, and when it comes time for the baby to be born, the muscle contracts, as you can see in this picture, and that is what pushes the baby out. When the mother is feeling contractions, it is her uterus squeezing the baby out until eventually the baby pops out. So just like there are relationships in organ systems of humans and complex animals, there are also relationships between the systems of plants. So I just wanted to provide the most obvious, easy to understand relationship, and that is between the root system and the vascular system that carries water up the stem, perhaps, to a leaf that uses that water for photosynthesis. So the root system, its function is to absorb the water. The vascular system's job is to carry water, and it also carries nutrients, but carries water to different parts of the plant, specifically to the leaves where it can be used for photosynthesis. Homeostasis is maintaining a stable internal environment. So basically we've got a variable, and I've provided a couple of examples in the next slide. But we have a variable, and if a change occurs and that variable becomes imbalanced, a sensor detects that. Then that sends information to our control center, which in most 99.9% of cases is the brain. The brain sends a message, which is an effector, which causes a change and allows that variable to become balanced. Negative feedback loops are what are used to maintain homeostasis. they will counteract a change. So if something becomes too high, a negative feedback loop will function to bring it back down, or if something comes too low, the negative feedback loop will function to get it back up. So I remember negative feedback loop, no, no, no. It doesn't want to change. Whereas positive feedback loops, these will encourage a change. So I remember that with go, go, go. So let's look at a couple of examples. So here are two examples of negative feedback loops. For maintaining homeostasis, thermoregulation, that is, our body maintains our temperature at 98.6 degrees Fahrenheit or about 37 degrees Celsius. So if the temperature starts to increase, okay, the stimulus is the increased body temperature. Our body's like, oh my gosh, our temperature is increasing. It will notify our brain, and then our brain will send out a signal to our sweat glands, and we'll start to sweat, and then the evaporation causes us to cool down. It'll send out a signal to our blood vessels, and the response will be that our blood vessels will dilate to allow the release of more heat. That will in turn cause our body temperature to decrease, and we'll be back at homeostasis. Now, if the situation were reversed, you're standing outside on a cold day, your body detects that it's cold. So the stimulus is your body temperature decreasing. That signal is sent to the brain, the control center, the sensor, and it will send out a signal to your blood vessels to constrict, and that will help conserve heat. It'll take your blood vessels away from the surface of your skin and therefore away from the cold. And you may also start to shiver. So a signal will be sent to your muscles to shake, and that will generate heat, and that will cause your body temperature to increase, and you'll be back at homeostasis. So this is a negative feedback loop. It counteracts change. Another example is blood glucose. So if there's an imbalance, if blood glucose goes too high, the pancreas will release insulin. That will go and bind to cells, and the cells will kind of open their doors, so to say, to the glucose, and that causes glucose to decrease, and you go back to normal levels of blood glucose. The liver can also take up some of that glucose and store it as glycogen for later use. If blood glucose level decreases, then glucagon is released, and that will cause the stored glycogen to be turned back into glucose, and now your glucose levels are increasing, and homeostasis is going back to normal, is being maintained. If this system right here fails, for example, the pancreas can't release insulin the way it's supposed to, the pancreatic cells are not functioning the way they're supposed to be, that results in diabetes, and that's very important. An example of a positive feedback loop is childbirth. So what happens is when the baby turns over in the mom's stomach, the head pushes on the cervix. This sends a signal to the brain. The brain then releases a hormone called oxytocin, okay? And then the oxytocin will cause the uterus to contract. And that will cause the baby to push more on the cervix, and that will send a signal, and then they'll release more oxytocin, and then it'll cause the uterine wall to contract more. That'll cause the baby. So you get you get the idea that, the positive feedback loop causes something to happen, which causes more of it to happen. So you just kind of go round and round and round and round. And the thing that will eventually stop is when the baby is born and no longer pushing on the cervix of the mother. And that should cover the review.